We have today the good fortune of having one of our members, Toni Pollin, who is a geneticist, a
genetic counselor,
researcher, and associate professor at University of Maryland at Baltimore. And I know that very well because I used to eat in their cafeteria when I was a social work student. So I know you.
Okay, so you know that campus.
I know.
Yeah. This little girl had diabetes since she was 15 months of age, and her mother had diabetes type 1 since 5 years of age. They were both on insulin pumps. Because of work that I did, which really wasn't discovery research, it was actually what we call implementation research. I was using information that I was looking for something that we already knew about. We found out that the cause of their diabetes was not what they thought, and they could actually be on oral medications. And it wasn't even like it's not like a new fancy expensive drug, which is probably why it's taking so long to get this into practice, because nobody's making money off of it. But they're on a drug that it's been around for centuries, and that directly targets the particular genetic thing they have. So
what does it mean that she's monochromatic?
Monogenic, yeah. So yeah, I'm going to talk about that more. First, I'm going to get a little bit more general, but when she said monogenic, it's because what I'll be talking about is that diabetes is. I'll talk more about diabetes in a bit, but diabetes is we've all heard of diabetes. But it's really not one disease. It's like many diseases. Some people think it's two diseases, but most diabetes, it's, people say is a genetic. It's influenced by genes, it's influenced by environment, it's influenced by a lot of stuff. But some people, a minority, but not nobody, obviously, have, unusual forms of diabetes that are due to very specific genetic variant, just the way cystic fibrosis and Tay-Sachs disease and sickle cell anemia are. But we're not looking for those. So a lot of what I'm focused on now, so to back up, because I did say I would talk a little bit more about who I am. I got into genetics because I always liked math. I liked math was like the class where it always had an answer. And
you were the one.
And I could do it, and I was good at it. But I also spent a lot of time as a teenager, and in college I did a lot of work with kids with disabilities, and at one point I majored in biology. I loved doing these fruit fly experiments, which some people think would be really boring, but basically we would to learn about how they used model organisms to map genes. We would look at these fruit flies under a microscope and look at what color eyes they were, and how many of them had red eyes and how many of them had white eyes, and then we would mate them in a little tube with food, and then we would count what their offspring.
Music.
What? Yeah. Well, just fun fact about genetics research that's not something you usually hear. So in order to look at them under the microscope, we had to etherize, we had to anesthetize them. But of course, we were going to want to mate them, so you didn't want to kill them. But the other thing was, it was not only was a delicate balance, because when you give them ether, if you give them too much ether, you might not kill them, but you might cause brain damage, and then they can't perform their mating rituals. I'm totally not making this up. And so, yeah, science. It's so much cooler than anybody. So
you give them a social drug? I mean,
yeah.
Viagra?
Yeah. Okay. Now, moving right along. So anyway, so I was interested in genetics, and I actually found it, it seemed tedious, but it was so amazing to be I'm counting these flies, and this is helping me recreate the process where they figured out that this gene is on this chromosome and this gene is next to it. And it was amazing, but I also really, I like working with people. So someone said, become a genetic counselor. So that's what I did. I went to do a master's degree in genetic counseling. And so what's genetic counseling? It's not like the DNA is lying on the couch and you counsel it, but it's,
that's a great That's a health thing.
But it's a field that really, it's a relatively new field. Which spoiler alert, I'm going to bring up a little bit of social justice in at the end, and so it's a relatively new field, but it's providing an increasingly vital service, which is, genetic counselors are not trained to in medicine to do physical exams, but they're health professionals whose training is how do I talk to how do I talk to people who have where something happening in their genes is affecting their health or their family's health or the risk of their family's health or their baby's health, and talking to them and help in ways that they can understand, and then also to be present with them as they make sometimes really difficult decisions. And so it really is a combination of genetics and counseling. And so that was really interesting to me, but I did a degree in it, and I really learned a lot and got a lot out of it, but I realized I just ultimately, multiple things happened. I worked in a prenatal setting, which wasn't necessarily the right fit for me, but also I just realized I wanted kind of more of the intellectual stimulation that came with like learning new things every day, instead of, because a lot of times once you're in clinic, like you do learn new things, but you're spending a lot of time with patients and billable hours and all that stuff.
And people are boring.
What?
People are boring. Genes are more interesting.
Well, what I also realized when I decided like I was going to switch and I was going to do computational genetics is that really good science now is working with people. And I think sometimes people come in now to the genetic counseling field and they say, well, I like science and I want to work with people. And I was like, well, just make sure that it's more than you want to work with people, because science is now because it's just so multidisciplinary. And so that's what I liked about it. I worked with different people, some people who were doing molecular stuff, some people were doing. So I was fortunate enough when I decided to switch over to research to land a job really 25 years ago as a research assistant with a group at the University of Maryland, where I still am, that was doing research on diabetes in the old order Amish. And then everybody asked me, why the old order Amish? Do they have a lot of diabetes? Well, no. One of the things we found out actually is that they're somewhat protected from type 2 diabetes, probably by their lifestyle. They still have a lot of it because it's a common disease. But they're a population that's been working with the genetics community since the 60s, because they're a closed founder population. They have a homogeneous lifestyle. They have lots and lots of kids. So if you're trying to follow all those mitochondria and genomes tracking, there's a lot of them. So I worked with that group, and so that got me kind of into the area of into really diabetes. And my work with that group was we were looking for we were recruiting lots and lots of people and their family members who had type 2 diabetes to try to find the genes that had small effects on whether or not people got diabetes. And we'd write these. I started getting involved with grant writing, and we write these grants like, oh, we're going to change medicine because we're going to find these genes that each one individually slightly influences. It's going to, but I went back to a lecture that I had when I was in my master's program about how some people have this type of diabetes called MODY. And so MODY was a term that was coined in the 60s and 70s when the two types of diabetes were known as juvenile diabetes and maturity onset diabetes. And these really smart people out in the Midwest saw these big families, and they were like, hey, wait a minute. These families have all these kids that have diabetes, but they don't have the same kind of diabetes that most kids have. They're not going into the hospital and immediately having to get get on insulin. It's sort of a milder diabetes. But they don't have the risk factors that you might think of for type 2 for maturity onset diabetes, like being overweight. Obviously they're not older, and also it seems to just look, when they get these families, the family tree, it was just looking like a single gene disease. And so that was in the 60s, 70s. It was in the 90s, which was right around when I was first starting to become a geneticist, that they actually found the genetic causes for most of these, and they're still finding more. So that's a little background. I wanted to just sort of show you. So this is a slide from a colleague, just reminding ourselves that I'm mostly talking about human genetics because that's what I do. I do humans, but DNA is the blueprint for all living things. So the way I think of Genesis, I think of is there was this one cell, or somehow there was the DNA got made into a cell, and then there was a cell, and then the cells started meeting up, and they're like, hey, let's form a community. And then eventually they formed, multi-cell organisms and then more complex organisms. And so that's where we find ourselves today. But to just talk a little bit about, I made this slide a long time ago. I just so DNA in context in humans, you could think, okay, So, you know, here's a person. And, you know, we have all these organs, we have brains, well, most of us. We have hearts, you know.
Some.
We have brains, we have hearts, we have pancreases. I had to put in the pancreas because I'm a diabetes person. You know, I'm skipping a few steps because the organs actually are composed of tissues and the tissues are composed of cells, but they have cells in them. They have these cells, and there's a lot of stuff inside the cells that make it work, but the thing that I'm most interested in is the DNA, which is on these, you could think of like books, like the chromosomes. And they, you know, certain times of cell dividing, they're like all wound up and then they unwind. And when it unwinds, you have this double helix that's made out of a sugar backbone, and it's got these, it's really let's see, Rabbi Emily said on the email the other day, something about it's like our ancient code. So I just was thinking a lot.
Okay.
What?
Was that wrong? Was that okay?
No, it is. And I was thinking a lot about how this is like, you know, it's like, oh, I'm presenting a synagogue. Oh, yeah, this is our ancient text. It's been copied and, you know, and some things have changed about it, you know, which maybe just like when the scribes, maybe some things changed. But, you know, just basically, you know, our cells so all of our organs and tissues are made of cells, and the cells have, the cells have a nucleus, and the nucleus has DNA, and the DNA is just basically a long code. It's a long list of A's, T's, C's, and G's. It's a book. It's a book that tells the human being like how to form, and then it gets. He talked about mRNA, so it gets changed into a message that can then be read by this machinery to make the proteins to make us who we are. And there's if you want to see more videos, you can probably see some more fun versions of this. And this is something that if you can actually apply chemicals to cells, and to actually I've done this. I did this with my own cells when I started the genetic counseling program, and you can and you apply certain chemicals and you can drop the cell and you can just see the DNA like all assorted into these chromosomes. And when you think of, and they're kind of like books, they're not, they each contain like, different genes. And then what we actually had to do to figure out like if there was any like major abnormalities is they actually, and people who do this for a living can like do this in their sleep. There's, this is pretty much what most people have is they have like people. I mean, animals have different numbers, but people will generally have 23 pairs of chromosomes. And you can actually see, when I said like, in the Bible, maybe if they thought like all the stuff, came from the dad, and then we're like, well, it's half and half. Well, part of the reason, maybe it's actually a little more from the mom is because, male babies get the smaller Y chromosome and whereas a female would have 2 of the X chromosomes. So Emily's like mind blown.
Don't mind, are those chromosomes, the 23, that's sort of what they each look like? They're each different?
Yeah, so what happens is when people are, like, one of the first genetic test there was like in the 60s, and that was where they could tell prenatally, like from a prenatal sample, if a baby had Down syndrome. And the way they could tell is because people with Down syndrome have an extra chromosome number 21. Most things that happen genetically aren't that dramatic. If you mess with the amount too much, usually it won't even, the individual wouldn't even be viable. But there are also these chemicals. They add this chemical called GEMSA that you would add to the preparation that would make the different patterns appear. Because you can see some of the 1 and 2, the first 2 pairs are similar size, but the people who do this all the time can tell the difference between those because they band differently based on the content, what content of A's, T's, C's, and G's they have. And then let's see, what else did I want to tell you? Oh,
so if you have identical twins, what's different?
Well, if they're identical, theoretically nothing. Theoretically, they will because what'll happen is the sperm and the egg will come together and create a zygote, like a single cell. And then when you get identical twins, it's because that single cell split into 2 individuals. And so they'll theoretically be identical. Now, the truth is, why do we have so much diversity among different species and among us? Because DNA doesn't always stay the same. It changes, it gets copied. Mistakes get made. There's things to repair those mistakes that don't cause too many problems, but sometimes they do cause problems. A lot of times they don't cause problems. They just change the sequence a little bit. And so over time, these changes can accumulate. And so so identical twins might have some differences because they might have had some mutations that occurred in their DNA after like they separated out and started dividing.
Sure.
So yeah, what?
But they start out.
They start identical. Yeah, they start out as the same. And this is just a little thing about that the guy in the video talked about, the mitochondria being the powerhouses of the cell. And so it's just he said something like that you only get, so you get, mostly you get half of your DNA from each parent, but you only get mitochondrial DNA, this special DNA that's in the powerhouses from your mom. He said that maybe because the sperm lack ambition or something, but I think what it is really is that the sperm are motile cells, so I think they need like the mitochondria. I think what I remember learning is that the mitochondria like go to the tail, like they need the mitochondria to like have the energy to swim around.
Does move?
Yeah,
Okay.
mobile, yeah. So but there's some stuff encoded in there, and it's actually interesting. It's if anyone's, I'm not really talking that much about that, but people who have done DNA for their ancestry, you can theoretically like trace back to the matriarchs using the mitochondria in similar ways that actually you can with the Y chromosome with the patriarchs. So anyway, so this is just this is what I mean. It's not really what it looks like, but it's how we configure it. It's a 4-letter alphabet, and it just looks like a random set of 4 letters. Like, how do you read that? How do you read that?
Very
So
carefully.
what?
Very carefully.
Yeah. Well, the cell reads it. We're actually, there's only about, I think for those of us that learned like DNA codes for proteins, only about 1% of all the 3 billion base pairs in our cells like actually codes for proteins.
Did you say 2 billion?
3 billion.
3
We have
billion.
3 billion base pairs. 3 billion base pairs. And most of it, only 1% actually makes proteins. The rest of it, we're still discovering what it does. It's regulatory, it's scaffolding, it's making these other molecules of RNA that never become DNA, and they just go and interfere with other things. So but that's what like a piece of DNA looks like if you just and I think I remember someone once saying, people think of biology, it's a soft science, but it's actually the only natural science with a digital code. So but this is
Wait, so it starts back, 99.9% of all humans have identical DNA, or something like that.
99. If you take any two people, about 99.9% of their DNA will be exactly the same. That 0.1%, if you do the math, which I'm not doing right now, but it's like 0.1% of 3 billion is still a lot, and so that's why there are differences between people, but compared to how much we're like, it's very small.
An aside, it makes it even more sad how much conflict and strife we have among us when we're
Absolutely. So this is what we know, what we understand most about how to read DNA. So the DNA makes this other molecule called RNA that's just kind of like the copy of it. And what happens is the whole alphabet when making proteins is read in 3-letter words. And there's like 20 different amino acids that make up all the proteins in our bodies. And so the way that the cellular machinery, the ribosome, like knows when it's making a protein, like what amino acid to add onto the string when it's making it is like, what is that codon? And also this AUG, not only does it code for an amino acid called methionine, but it also it's known as the start codon. So if you interfere with it, then it gets all messed up because it's like, I don't know where to start. And then, this one codes for like threonine, aspartic acid. And then there are a few codons that code for stop. And
what?
so
For what?
Stop codon, yeah. So it's like reading along, it's like I'm stringing together my amino acids to make it's like a polypeptide, it's a string. Eventually then the polypeptide like folds into different things to become proteins. But when it comes to that stop codon, it's like, okay, I'm gonna stop. and that's how it knows to stop.
To stop what?
To stop,
dividing?
like, yeah, they'll like, you're done making that, yeah, you're done making that protein, stop.
The car's ready.
Yeah, yeah. Well, they tell me that sometimes, and then the car's still up on the ceiling.
They're not geneticists.
Yeah. And then, this is like the code that it's reading. And so you can see that there's only four letters that form 3-letter words. And it's redundant too. So that's the other thing that kind of protects us, even though we have mutations occurring all the time. One of the things that protects us is a lot of it's not coding for protein, but one of the things that protects us is that you can actually change the codon that and still add on the same amino acid, so that it won't usually make that much of a difference. So, what else did I want? Oh, and this is a slide that this is happened to like. The only reason I wanted to show this is because sometimes you think, well, why? Because we have the same DNA in all of our cells, right? So why don't we just all look the same? And that it all has to do with things. First of all, I'm not a developmental biologist, so I'm going to do some hand waving. But really, there's all these things that happen, like signaling of what position. Like in the beginning, you have its embryo, and then it starts as a ball, but then it starts to like have, a top and a bottom, and there's all this signaling that happens because of those positions, and then eventually the micro environment or the environment that like tells it what's going on. So this is just an example of some of your DNA, we call the part that's the protein, the parts in red, the gene. That's kind of the conventional definition of a gene, although that can be changing the more we understand. And then what happens is it's not like every gene makes a protein in every cell all the time. There's regulatory things going on. And so this is just an example showing where there's a glucocorticoid receptor, and it binds to a certain point upstream on the DNA, upstream of where the gene is supposed to get made into a message, it's only going to result in the expression if there's glucocorticoid hormone to bind to that receptor. So I hope that I think I lost everybody.
So it's
But-
nice
have glazed over.
glucocorticoid, I just hear the word.
Forget the word glucocorticoid.
Okay, no, I was wondering if that's a tie into diabetes. Because
No, it just
it's
happened to be what this
Okay,
example is. It's
okay.
just that there's like stuff around that affects whether certain proteins can like stick to the DNA and then tell the DNA like make some more of that gene. So it's just that it's complex. This is just an example of that. So that's why it's not like, that's why I just to get some sense of why we're just not like a bunch of identical cells, because there's so many different things going on. This is a nice little kind of timeline of genetics from 1865, and Gregor Mendel. Do you guys know the name Gregor
Sure,
Mendel?
Yeah.
When he planted his peas, he was a monk,
Peas.
and he had, he figured out something. Darwin was around then. Darwin was like, figuring out evolution. Mendel, they didn't know what DNA, but he was able to observe in the plants what's really formed the basis of what we know about inheritance today. I think he kind of ignored some of the examples that were a little too complex for some of the things we know now, but we didn't know. But and so so I'm not going to go through this whole thing, but I guess one of the things I wanted to ask was, how many people have heard of Watson and Crick?
Yeah.
How many people have heard of Rosalind Franklin? Okay, so more people have heard of Watson and Crick than Rosalind Franklin. So who were Watson and Crick?
Supposedly the people who discovered the double helix.
Uh-huh. And who was Rosalind Franklin?
She was the one who really discovered it.
Yeah. So
Yep.
So-
That's
so
why
a
I said-
Passover, yeah,
That's why I said supposedly.
supposedly.
Yeah.
And they did their work was important. It was theoretical, but so on Passover I wore a dress with locusts. So today I'm wearing this. This is a reproduction of the very first photograph taken of DNA by Rosalind Franklin.
Oh, that's so cool.
Oh, that's
Rosalind Franklin, not only she was a woman, she was a Jewish woman from England. And some people think, oh, she didn't get the Nobel Prize because she was a woman. Well, it was complicated. I mean, basically what happened was, it's a really interesting person to read about. Like, she somehow made it, in the sciences. I think, she came from a family where there was money, but she wasn't allowed to eat in the cafeteria with the male scientists. And so people thought of her as like standoffish, but like she wasn't allowed to. And but the story is that Maurice Wilkins, who shared the Nobel Prize with, like, you know, secretly shared this picture that she'd taken, and and they had been doing really important theoretical work and building models, but without that picture, then that, you know, that was the proof.
Wait, the picture that's on your dress.
Picture, because it shows, it's an X-ray micrograph of the double helical structure of DNA. It was the first.
So Toni,
Yeah.
how do you wind up with a piece of clothing that has
Amazon. No, not Amazon. I really should start getting kickbacks from this company. It's called savahausa.com.
Cool.
And it's like it's you know, they've grown, but it's like these two women who were just like, they wanted to like kind of overcome stereotypes about scientists. So they make all these beautiful like science,
so cool.
technology, engineering, arts, and mathematic themed clothing. Yes.
Awesome.
So yeah.
Very cool.
So that was 1953 was kind of when it was like discovered, like DNA it's a double helix, it's the source of life, how does it work? 50 years later, 2003, which was like while I was working on my PhD, was like when the first like human genome sequence came out. And it was an interesting time because it was like a long time project of the NIH, which James Watson, who I think is still alive, was involved in. He was the first director, I think. And then like this commercial company got involved and probably sped it up because then it became this big competition. And so they both the commercial genome and the government genome came out like the same time in the two top journals, Nature and Science. And I still remember there was like a picture of Francis Collins and Craig Venter like on the front page of the Washington Post, like, you know, standing there smiling together. So now it's been 21 years since then. So there was already a lot of some genetic discoveries being made then. I mean, you could already, people who are familiar with Tay-Sachs screening, that was even, they didn't even have the gene when they were doing it because they could do it by the amount of the enzyme. But now we are now at the point where it's becoming almost routine to do a person's genome in like less than a day and like actually even interpret it like in like 5 days. So things have moved along really quickly. There's newer technologies okay. So that's a little bit about DNA. What did I have here? Oh, I just sort of want to show you that, you know, there's all different tools now that we use in genetics to make sense of, you know, and this really is, it's like, it's, you know, I thought that another project I'll tell you about it's very much like we have the text, and actually we don't have the full text. Now there's something called the metagenome. There's more and more efforts to sequence more and more genomes from more and more populations because the original human genome was based on like 5 people. And so now there's more effort to get more to understand the full genomic diversity. But you know what we're tasked with is really making sense of an ancient text. So that's I just attended a meeting yesterday and Thursday. It was actually it was this year. It was in Baltimore, called Curating the Clinical Genome, and the whole meeting was about groups that are about basically curating what does it mean? What does it mean with respect to disease? How do we talk about it? How do we curate it? How do we put it in databases? How do we make it accessible? How do we so I just thought I'd show you this. There's a website called OMIM, which stands for Online Mendelian Inheritance in Man. It was started by Victor McCusick, who's sort of considered he was at Hopkins. He's kind of considered like one of the, if not the grandfather of like medical genetics. And in the 60s, he was writing books. Every year he'd write a new book, Mendelian Inheritance in Man. It was just like a catalog of every trait that looked genetic. I mean, because he didn't have, the DNA tools, just every trait just looked genetic. Yeah. He did a lot of stuff with the Amish because he recognized that these isolated populations you could, learn a lot from. And so at some point, probably shortly before, sometime in the 90s, it became an online resource. And so I just kind of pulled it up today. It says there's total number of phenotypes for which the molecular basis is known, 7,530. Some of these they're not all diseases. Some of them are just traits. Total number of genes with phenotype causing mutation, 4,910. So
And
there's
this will just continue to grow.
and this is going to
right?
continue to grow. Yeah. So there's just a lot of knowledge, and one of the challenges right now is we need to figure out better ways of getting it
Is
into the
AI
hands.
going to be helpful?
What?
As AI?
Yeah, I mean
continues to develop,
AI.
will
I
that
mean the
be
way I think
helpful?
of AI is it's part of it. Yeah, but it's to me the way AI works is you figure out how to do more and more complex things, then you teach
And then you
AI how to
teach
do it,
Right.
and then you can do other things.
Right,
And then
exactly.
you just keep
Yep.
So that's and
So you said this is interpreting ancient texts,
Mhm.
right? So it's like Torah study.
yeah, that's well that's what I realized when in part because of what exactly no I realized how much in common that it has because it's
Do you take your microscopes once a year and like dance around the room?
I feel like this is a deeper, like not saying anything about your particular Torah study, but than any Torah study, because I'm like in awe of the universe on a deeper
Yeah.
level than I ever could be,
Yeah.
Anyway, just the complexity of it.
It's different, yeah. So this is another tool we use, and I just thought it'd be fun to show because there's something called NOMAD. It's an acronym, but it has more and more. It has 800. It has genomic either the genome. So a genome is all the DNA, or the exome. That's that meaty part that codes for the genes. From now has 807,162 individuals, and this is really important because one of the things we need to do is when we're looking at an individual person's genome who maybe has a disease, and we say, oh, we find this variation. Well, we need to know is one of the things that helps us figure out if this variation is causing their disease is this found in the general population, and how commonly in the general population. But what's the general population? And historically, like a lot of other things, there's most of what we know about the general population has been from white European individuals, and so there's more and more efforts to get more diversity into the tools. And I just said there's the new version now has actual labeled Middle Eastern genomes specifically for the first time, but you can see kind of how many different and Ashkenazi Jewish is a specific group that's listed, but you can see just like how much DNA you need from like individuals, to make sense out of, it's just all mind
Well, to
boggling.
talk about the general population.
Yeah, what is the general population? Yeah.
So, I was speaking of, saying Jews and the Amish, since the Orthodox can be very, very much their own community and insulated, have there been any studies of?
Yeah, and I know some of them. There are people in Israel and New York who've done a lot of studies with the Ashkenazi Jewish population. Yeah, and depending whether you're talking about the more insular ones or the, there's different commonalities and differences. One of the things that's really interesting, I don't know if I think some people probably here are aware of like carrier screening. Now it's becoming more of a universal thing where people are pregnant and they want to, but it used to be very specific, historically specific carrier screening for people who are Ashkenazi Jews. But there's a whole, how do they screen for Tay-Sachs in the ultra Orthodox? I don't know if anyone knows this, but it's not like people just get genotyped. It's like there's somebody who keeps, because there's so much stigma around genetic conditions that they get like genotyped in high school, and then somebody like owns the database, and then when a match is being proposed, then they try to like avoid matches that would and then they don't have to deal with because then once
So do those people, is it hard for them to get married because of that?
Well,
Who
the
knows?
thing is, it becomes hard for the more things we look for, and that's. I was actually part of a conference that an Israeli genetic counselor I know did at Sinai, sort of about like removing stigma, because basically like we all have we all have stuff.
Yeah,
But-
my uncle, who was a breast cancer expert, he actually did a major study just in Israel because, breast cancer, is predominant
Yeah.
in Ashkenazi Jewish women. And so he went to Israel and basically got tissue samples from over 100,000 women, right, in order to do studies
Yeah,
on
and
breast
that's why-
cancer. And he was one of the people who identified that there's not just one type of breast cancer is not one thing, and he was the first one to say, let's stop with these radical mastectomies right away. And
Right.
Yeah.
Yeah. So, I talked about this already, but this is like the picture from an Amish quilt auction, which just to mention that I had worked with the Amish, and I know we're short on, I don't know if, but I just wanted to kind of highlight, so I mentioned that, the work group what I started doing when I came to the University of Maryland was working with this group that was working with the Amish. Unlike Victor McCusick, who was studying rare diseases, we were studying common diseases, and my original division chair was Alan Schuldiner. He was a diabetologist, and he wanted to kind of reduce the complexity of understanding the genetics of type 2 diabetes by studying the Amish, because they have both like a smaller gene pool and also like a more homogeneous lifestyle. And so this ended up growing into, now we've got a clinic up there. We've recruited like over 10,000 people. We have DNA from over 8,000 people from the Amish. And so found a lot of things, and I'm just gonna skip right to the highlight, to kind of my proudest accomplishment in working with the Amish, is that we worked on a study called the Happy Heart Study, where we were looking at, it was heredity and phenotype intervention study. So we were looking at like short-term interventions that were related to cardiovascular disease and what are the genetics that affect those. So one of them this is really serious. At hour 0, we gave everybody a very high-fat milkshake that was adjusted to their body weight, and then we measured their triglycerides for 6 hours after they drank the milkshake. And what we found is that what this is showing here is that some people, is that, generally our triglycerides, because our triglycerides go way up, like during the sitting when we're drinking fat, but some people didn't go up as much. And it turns out that they actually have basically essentially like a one-letter misspelling that causes the stop codon to be too soon. And so they only make one copy of this protein, or half of the normal amount of this protein, called ApoC3, which is a protein that's bound up with triglycerides. So if you have less of it, you actually have less triglycerides in your blood. And this turns out to be seems to be cardioprotective. And they actually had less coronary calcification. And then later, some people, once it got easier to study these rare variants in bigger groups, basically showed pretty convincingly that having this reduces the chance of a heart attack by like 40%.
Wow.
And good news, I found out that, because I eventually got my genome sequence, I found out I have a variant, not the same variant, but another variant with a similar effect. And it's found in 1 in 42 Jews.
Are you partly Amish?
No, it's found in 1 in 42 Jews. So I was like, oh, maybe that's why I got so interested in it. So, but I don't know, for those people that just sort of this is like the part of it's a pretty small gene. This is the part that actually codes for the protein.
So is part of your work maybe eventually to be able to make that change in people that don't have it?
Well, not my work, but there are actually multiple companies that are like stage 3 that are publishing now clinical trials where they are doing that. They're like giving, and this ended up being, it didn't make any profit off of it, but this finding, it matched with some stuff that was known, but kind of mainly for mice. This was like the biggest population where this had been seen, and it basically provided the data that said, hey, you can have half the amount of this, and it seems to be safe, and that helped to kind of, actually, it's funny, at one point I gave a talk on this to a company that is now called Ionis Pharmaceuticals, that they were all about like making these molecules that like basically like destroy the message so that you don't make as much. And they used to be called ISIS Pharmaceuticals. And you probably figured out why they changed their names. Yeah. But anyway,
You mean ISIS is into that also?
Well, ISIS Pharmaceuticals. So, but anyway, this is just the thing that I had made a long time ago to show like, how basically this just like, because it changes a C to a T, then it changes something that's supposed to make them, the arginine, instead of making an arginine very early in the protein, it's just stopping it. So it's just like not making anything. Also, this was fun. I dug this up, that I hadn't looked at this for a while, but I actually like figured out, I because we have these Amish family trees, pedigrees, and I was actually able to trace it back I think it came into the Amish. It didn't happen in the Amish because we've found it in other populations, but I think there was only one copy that came into the Amish. It's because it's relatively rare in the rest of the population. And then it propagated through these large families that they had. Wow. And I actually looked up in the genealogy book. We use these genealogy books with a computerized database that we work with NIH. And I think it's him. I think we don't know which one it came from, but I think his name was Shem Esh.
Oh my goodness.
So I just had to show that here.
Wow.
Okay, in the remaining time, I'll just like switch to diabetes. So, this.
I'm
Yes.
sorry, just reading.
It's okay. Yeah. So basically, I remember I saw this great talk once about how, basically diabetes, there's lots of different ways you can mess up blood sugar, because blood sugar is energy. Like it's how we're alive. So it has to stay like in a tight interval, and there's lots of things that can knock it off. You can tolerate it, not like, so a lot of people get diagnosed with diabetes like without any symptoms, but they should do something about it because they could get sicker. But just, this is just like a little mini thing about what diabetes is. It's like, what's supposed to happen is like the pancreas is made of lots of cells, and some of them are beta cells, and they make insulin. Make insulin when there's a lot of glucose in the blood, and that like induces the liver to store the glucose as glycogen so that it'll keep the glucose levels in the blood down. And then there are these like other hormones made by other cells in the pancreas, like that make hormones like glucagon that actually kind of counteract that, and that's why like you're able to fast and not like completely have your blood sugar go down. And so when this goes wrong, it could be for a few reasons, and there's been many different ways that diabetes is classified. This is how it's currently classified. So type 1 diabetes is, if it's really type 1 diabetes, it's an autoimmune disease like MS or Crohn's disease, like basically the immune system is attacking the beta cells. But sometimes we assume that's what it is when that's what it is. Type 2 diabetes, people think of as a disease, but those of my cohort, we call it a diagnosis of exclusion. People think of it, oh, it's a lifestyle disease. No, it's not a lifestyle disease. You're more likely to get type 2 diabetes if, as people get gain more weight and are more sedentary and are older, because those all like reduce the ability of the body's tissues to respond to insulin. But there's also lots of genetics going on. And then there's these other specific types, so I always like to be on the margin, so that's what I'm most interested in. And
of course,
gestational diabetes, and people think, oh, monogenic diabetes. That's what the mom in the second movie was talking about. Oh, it's rare, but it's like 1 in 100. Like the conservative estimate is about 1 in 250 diabetes cases. That's not
so rare.
Yeah,
Right.
And so this is just.
How does genetics correlate with hormones? Because like they say, if your cortisol is off, you can get diabetes or become overweight. Is there any correlation at all?
Yes, I mean, yeah.
From a trauma perspective, and I defer to you because I'm not.
Yeah. No, I defer. Yes.
So
on trauma, I defer to you.
When there is more cortisol in the system, the fight or flight response includes increasing the blood sugar. So it's muscle tone, heart rate, your cholesterol goes up when you're in fight or flight. And ideally, the fight or flight cycle completes and the body discharges the chemicals, but that's generally not what happens. And so when people have very high cortisol levels, that's a lot of moving parts in and of itself, I think it probably could contribute to.
Right, and then I think what could happen is where it can affect the genetics is, first of all, your genetics can affect your response to trauma. Mm-hmm. So trauma isn't something that happens. It's so different bodies have different chemical makeups, and then once you have that cortisol, then what's going on with your metabolic machinery can affect how much that cortisol change will affect. So yeah. And that's just very lot of hand waving, because that's all like, like, I mean, yeah, I mean, because it's all a bunch of little specific things that you have.
Yeah,
people,
I just gave my
yeah.
full extent of knowledge on cortisol and
That was
Yeah, no, but I mean, that's,
And it was
but yeah, but that's,
understandable.
yeah, but, and then, but there's so many, all these little things that happen before the cortisol and after the cortisol, and yeah. So so that's, so was that kind of your question? Yeah. So I don't know, I made this a long time ago, before we even really had many type 2 diabetes genes, but, because people would be like, why do you study the genes of type 2 diabetes? It's a lifestyle disease. I'm like, no, it's got some genetics involved. And then they'd say, well, it's not about, like, isn't it caused by, like, obesity? I'm like, well, yeah, obesity is a risk factor. And people kind of generally think of obesity as like a lifestyle risk factor, like it's an imbalance of calories coming in and calories going out. But really genes, we knew just from studying families, and now we know because we got the genes, that genes actually influence body weight. And part of how they do it is through these things, through appetite, through food preference, through, like, my sister who loves marathons, and I don't, because of, that's got to be something partly genetic. So and then type 2 diabetes, it's like I always remind people, diabetes isn't really an endpoint, because it's the complications, the things it does to your eyes and ears and blood vessels, and how, what controls that? Well, how well are you controlling it with medication? How well are you controlling it with all these other things? And then
you study pre-diabetes.
Yes, yeah, and pre-diabetes only became a term after, like, in the early 2000s, there was a clinical trial called the Diabetes Prevention Program, where they quantified the extent to which lifestyle interventions and medications could slow the onset of diabetes in people who had what used to be called impaired glucose tolerance. But they started calling it pre-diabetes because of that trial, because then it became important for doctors to, because there was an intervention that was evidence-based. But anyway, and then I always just like to remind people again that we kind of talked about with the glucocorticoids, when I told you to ignore the glucocorticoids, like, the stuff that happens to us and it affects how the genes get expressed. It's not, we think of it as a one-way. The central dogma goes like this, but it actually goes like this. So then, but there was this big boom in diabetes genetics when this ability to put like thousands and thousands of common genetic variants on a chip came in like 2007. And so then now we have like over 200, maybe not, I don't know, this is already dated, maybe I have over 400 areas of the genome that impact, but mostly in small ways. These are like what are called odds ratios, so like 1 point something fold difference, whether people get type 2 diabetes in conjunction with their cortisol and all the other things. But what I was really interested, I started seeing papers like this when I first started working in diabetes, where people had a genetic diagnosis of HNF1 alpha diabetes, alters treatment and improves glycemic control in the majority of insulin-treated patients. So there were patients who were diagnosed with type 1 diabetes, put on insulin, which is the standard, like they're assumed not to make insulin. But then, like, it was discovered that some people have this type of diabetes, that MODY that I mentioned, and it's actually due to a mutation in a gene that makes something called a transcription factor that's like in the nucleus of the pancreatic beta cells, and it like tells other genes, like it regulates other genes. Basically what they found was that most of these patients could switch to these oral medications, low doses, actually lower than normal doses of oral medications that were originally used for type 2 diabetes, and control their diabetes, and often have better control. And this especially just related me back to my genetic counseling background, because sometimes I wonder, well, I'm going into like, quantitative, like nerdy stuff, like, what is my genetic counseling background relevant? But this really got to me, which said the people who went through this, some of them said, I don't feel like a diabetic anymore, because their life changed so much, because they were no longer like tied to a pump. This is a fun little schematic of a beta cell, and this is just showing kind of I mentioned that the 3 genes involved in these monogenic forms of diabetes, MODY, were found in the 90s. And then other ones later were found in like the 2000s. And I don't know how well you can see this, but, basically the really kind of practical implications of this was that people who had one form of these, the ones where there was something going on in the nucleus, they could be treated with these low doses of that oral medication. There's another type where they have an enzymatic problem where their fasting glucose is just high, slightly high their whole lives. But because you might have a routine exam, like a kid breaks their arm and they go into the doctor's office and the doctor, and their glucose comes back high, then they might think, oh, this kid's at the beginning of developing, type 1 diabetes, better put them on insulin. And in fact, this is actually one of the hardest ones we find when we're trying to sort of convince doctors, it's hard to you can maybe convince people treat this differently, but to convince people don't treat it, and the numbers keep showing up, because the numbers won't change because it's just normal for them, but it's just a hard thing to grasp. And then there's this other form that has to do with this whole potassium channel, and that's the form that Ava and her mother had.
Same doctors feel they need to do something.
What?
Sorry.
How does the doctors feel if they have to do something?
Right.
That's
Yeah,
what
no,
doctors do.
it's much harder to, yeah, exactly. And so, and we're very numbers driven. So these actually, after the first, so I did a study basically where the study was an implementation study. So it's like, okay, what happens if we just look carefully at people who are seen in a diabetes clinic to see if they might have these features, like they were diagnosed really young, they don't have the antibodies for type 1. It actually turned out to be a national study, because even though this was sort of supposed to be standard of care, it wasn't being diagnosed, and so we had people flying from other parts of the country paying their own money because we didn't have money to pay them. But these are a couple of the patients we diagnosed at the beginning, and these were actually from the Baltimore Sun, and they both. This one was a young woman who, when she was in college and then her late 20s, she was told, you don't have she didn't have type 1 diabetes, but you have your glucose is high. You better do something about your lifestyle. And she's like, I'm a kickboxer. I train very hard and I hardly eat any carbs. And so she just knew something was wrong. And she actually got us on the front page of the Baltimore Sun in 2015, I think, because she couldn't come. We tried to, she couldn't come when the Baltimore Sun came to interview us and take pictures. And so they called her and they went to visit her at her studio. And then this woman actually, she had a diagnosis actually of type 2 diabetes. She was being treated for it. And her kids a couple times again, showed up at urgent care and happened to have their glucose measured, and it was a little bit high. And then it just started not to add up, and then they figured out. And so they both have that one form that doesn't need treatment called GCK diabetes.
So do you have anything in the works, these drugs now, Ozempic and whatever? I mean, it's selling off the shelf. Any studies?
I'm not working on that. No, it's not
Is
in
there
my
such a
purview.
thing? I just didn't know if they correlate to genetic, because supposedly you lose weight and theoretically you lose weight.
Yeah.
There's less type 2 diabetes.
Yeah.
in
Well,
theory.
but again, I'm working more on these types of diabetes where that's really less the issue.
Not the
Yeah.
implant.
But
And Alan, by the way, the weight loss is secondary. So it was started as a drug for diabetes to lower blood sugar and A1C, and people were losing weight, which is why it's so popular. Right?
Yeah.
Viagra's heart medicine.
Right.
People were taking heart medicine, they started to grow hair. So there are a lot of people on Mounjaro, Ozempic, and those drugs whose sugar goes down and who do not necessarily lose a significant amount of weight. That's how you hear about it, because that's what's popularized it.
Yeah.
Right.
well, and to be fair, sort of to how the medical community sees it. Losing weight loss improves health.
Right.
And so there's a certain amount of weight loss that has to happen. And so doctors are excited about it, because even though, yes, you work on lifestyle, and some of the lifestyle stuff I think is structural and things that need to happen, not just telling, you can have a whole another discussion about that. But the reason people are excited about it is it does enable the kind of weight loss that can make a difference to health. But, of course, any weight loss drug, then there's a whole another culture around it. But I just want to just have a couple more things to talk about. What happened when I was doing this project where we identified, Ava's genetic variant at the beginning and their genetic variant, is what I realized is that, you have all these different variations. Each gene can have thousands of different variants, and some of them are just normal variation, and some of them cause disease. And so this one thing led to another, and I ended up forming, and this is a major part of my work, is that I run this international group of experts from all over the world, and we have calls every week. And it's and coming back to the Torah thing, the discussions we have about like whether this variant causes diabetes and whether this warrants using this criteria, it feels very Talmudic. And it's very fun. And so this was actually our last meeting before COVID, where we had like 15 people from 8 countries. But this kind of shows the countries where people are, and it's you know, it's very much like the blind man and the elephant, because we've got people, because it's like two things. It's people sharing different, there's so many different kinds of expertise. You need the clinical expertise, the medical expertise, the science, people who can like put these mutations in cells and figure out what they do. But then also it's like, so Ava's mutation that we found, that mutation had been found in one other patient, and it was reported in the literature. And if it hadn't been, we wouldn't have been able to report to her, and we wouldn't have been able to help her diabetes group, like switch her from so it was very sobering. So realizing like we've got to get all this stuff together, because it's just the nature of these they're rare, but there could be 10 copies in the whole world, and then once you have 10 copies,
This
then you
kind
know
of
what they do.
world coordination and conferencing is in itself, I think, fairly new, right? It helped take off during COVID and
yeah.
yeah.
I mean, we were doing that before, but certainly the tools that the availability, the improvements, I think, in the teleconferencing tools that came about with COVID makes it even easier. And then this is actually another highly collaborative study. It's a national study where this is people who have diabetes that doesn't look like normal diabetes, like doesn't look like, well, it's atypical, which, what does it mean, atypical? We're all atypical, right? But there's like, it's like, I know it's, so it's, even that's a complicated thing, but people who might look like they have one of these unusual forms with their genetic testing is negative, we actually have a study to look at them. And then the last thing I wanted to say is, so I mentioned genetic counseling. This is there is a definition on the web, but it's less concise than this. But I'm going to end with just like some activism. Because genetic counseling is a relatively new profession, and just a fun fact, genetic counseling is a covered service by reimbursable service by Centers for Medicare and Medicaid Services. Genetic counseling, but genetic counselors are not. So a doctor can do it, a nurse practitioner can do it. It's because genetic because genetic counseling is a new profession, and it never. In part because it was started by some like people who were like, I don't know, who were just motivated by other things and so it's not yet recognized as a profession, as a healthcare professional by CMS, but genetic counseling is recognized. But genetic counselors are the only people who actually receive consistently formal training in how to do it. And so we actually recently got licensure in Maryland. Not all states have licensure, which will help. But in order, and it's covered like if you have private insurance. But in order to get equitable coverage, so that like, because CMS sets the standard, we've been trying to pass some legislation, and I just happen to have a QR code that makes it really easy to, and I can send the link too. It's a complicated link, so I didn't put up the link, but basically, I think I haven't even really read all the bills, but this says it all. It's like, this is the whole bill. Go and learn it. And it says, this bill provides for coverage under Medicare of genetic counseling services that are furnished by genetic counselors. Covered services include those services as well as incidental services and supplies that would otherwise be covered under Medicare if provided by a physician. So
that's really complex.
I know.
I don't know if senators and congressmen will
and this is like, I think this is the third incarnation of the bill. It's had other numbers and stuff. And I'm probably like, I am a genetic counselor, but I probably like, that's what genetic counselors are really trained to do, to be able to talk in ways that people understand and to help them, through. And there are a growing number. And I think we will be able to have more access to them anyway. So that's my okay, stepping off my soapbox. Oh, well, I mean, a lot of this was a soapbox.
This is great.
So anyway.
Thank you.
You're welcome.
Do we have time for a couple questions?
I do. I mean,
So
if people have to
Well,
go, then I
actually,
understand.
I don't have any problem. I probably shouldn't go more than another 10 minutes.
What
let's
time
say.
is it, please?
It's 2:10.
7 every, yeah.
Okay.
So
Okay, I've got a hopefully a quick question.
Yeah.
Is it because the genome that impacts on diabetes is so complex that they can't think about the gene splicing stuff that you hear about? Where they
Oh, you're talking
correct
about
it?
gene therapy. Yeah, I did skip a whole bunch of stuff. Yeah, a whole breakthrough thing. I mean, another just important like highlight of genetics today is easy to understand. There's a disorder called spinal muscular atrophy. This is like this motor neuron disease where the babies with the most severe form, like they lose the ability to walk and they die they did die by the time they're 2. There is now actually a therapy that literally changes the DNA, and these babies are like literally
One of my
walking.
neighbors had that.
What?
One of my neighbors had
Oh, they did.
Wow.
So they had did they have the therapy?
Yep.
Okay, yeah. So that's really important. So I mean, I think for a while, gene therapy was difficult because like you have to figure out how to get it in, and it can cause off-target effects. It's improving. So there's a lot of this is a heyday for gene-based treatments. That not just look at the pathways, but actually like replace, and it depends on the tissue. Like we're never going to be able to cure everything, and then it introduces all these ethical issues about germline manipulation and stuff. As far as diabetes, I think in part, it's funny because it's actually hard to. It's like there is sometimes the priority is where there's not a treatment, and I mean people live with diabetes. And in fact, it's often even hard to get physicians to be like, oh, consider that this patient might have MODY. And the patient might not, especially because testing has been expensive. I mean, that's a whole part that I didn't go into, and it's gotten cheaper now.
Sorry, what is MODY again?
Well, it's a type of monogenic, single gene diabetes.
Okay.
And so, you have someone who's on their insulin pump, they're living their life with diabetes. And you say to them, well, this genetic test could show that you have this other thing that could be treated with oral medication. And then anybody like, well, I have to pay for this and I'm doing fine. So I don't know. So that may be just, it's not really, I think for most, maybe for some of the, like there are some neonatal forms of diabetes, like that one that's mild. If you have 2 copies of it, then you can't make any insulin. But I mean, you think about what's really fascinating, I think, is insulin insulin is just a little over 100 years old. And that was a breakthrough because before that, people with, basically they would just start, like kids with diabetes would just, starve. That was how they prolonged their life by like just feeding them less. And then, what's amazing about that is like without the internet in 1921, it like went all over the world. I mean, not all over the world because, obviously there was not equity and access, but people all over the world were using this treatment, when they had to extract it from like slaughterhouse, pancreas and stuff, pancreases. But yeah, so it's, yeah, I don't know, diabetes, there's not, it's not, the ones that are really, I think, being looked at with some of these like really cutting edge therapies are like the like fatal conditions and not so much like chronic diseases. So that's a long answer to a short question.
Okay, one more question.
Okay. Okay, let's say if I can't think of anything right now, meet someone with diabetes or find out someone I know has diabetes, is there anything or suspects they do, is there anything I can tell them or maybe even tell them to like go to their doctor and ask to be tested for X or anything like that?
Well, I would say, so yeah, that's my next project is to like create an infrastructure where people can get access to that information. But I think, I don't know, I think one of the things I've learned is regardless of whether you're talking about rare types of diabetes or common types of diabetes, sometimes people, like, it turns out type 1 diabetes, the autoimmune diabetes, can occur at like any age. And so sometimes
It's not
adults,
just young.
yeah.
Really?
So sometimes,
In my 50s.
Wow.
you got it
Wow.
in your 50s? Yeah.
Really?
Wow.
Yeah.
And sometimes adults, especially if they're like overweight or obese, are assumed that, oh, you must have type 2, and they don't even they try lifestyle and try this, but I think and children too often sometimes if so basically testing for the antibodies is really important, and then it's a matter of looking at whether people have other risk factors, because that's where we discover it the most. Like I mentioned, this kickboxer, like she clearly she was in kickboxer in her 20s, she clearly didn't have type 2 diabetes, but but then somebody might have both, they
like
might
Mark
have
Andrews has
weight
diabetes. He's our tight end for the
Tight end,
Ravens.
yeah.
Yeah.
So yeah, and I don't know if he has type 1 or
Type 1.
yeah, and of course, again, those numbers are. So I think where it becomes an issue is if something isn't working, something doesn't seem normal or something doesn't seem. A lot of times if there's family history, but there's not really one thing. But you can always just, if he has questions that he thinks he's atypical, just send them to me.
Thank you.
Well, thank you, Toni.
Thank you
so
so much.
Thank you.
So much more I could have talked about.
Well,
We
we can have you
know.
back next
Oh.
year.